We've all heard the saying, "What doesn't kill us makes us stronger." But who could have imagined that millions of years ago, a single moment in time would be responsible for shaping an entire planet and its inhabitants? That's exactly what happened when an event threatened to destroy every living thing in our now blue-green world as we know it. In fact, it was so powerful that its traces stayed within our DNA. Hello and welcome to Curious Media. In today's video, we will be looking at the pandemic that lasted 15 million years. But before we begin, don't forget to subscribe to the channel and click the notification bell for more amazing content, all about the ancient pandemic. Scientists have found an ancient group of retroviruses that spread around the world and infected the ancestors of modern mammals about 15 to 30 million years ago. Actually, if you look at it, retroviruses are fairly common in nature and include a few common ones like the human immunodeficiency viruses HIV-1 and HIV-2, as well as human T-cell leukemia viruses. The research on a group of viruses called ERV-FT showed that they affected a wide range of hosts, including carnivores, rodents, and primates. The fact that ERV-FC is found in these very ancient mammals suggests that the viruses spread to every continent except Antarctica and Australia and jumped from one species to another more than 20 times. Apart from that, scientists also said that the study shows that ERV-FC has been around since at least the beginning of the Oligocene epoch. The Oligocene was a time of big changes around the world, including a cooling of the climate that led to the ice ages. In fact, around this time, vast swaths of grassland emerged, along with large mammals as the world's dominant fauna. Actually, viruses have been around for billions of years and can be found anywhere there is life. As a result, they have a large impact on the ecology and evolution of all organisms, from bacteria to humans. But unfortunately, viruses don't leave fossils behind, so we don't know much about where they came from and how they changed over time. Over millions of years, however, viral genetic sequences have built up in the DNA genomes of living beings, including humans. These viral genetic sequences can be used as molecular fossils to learn about the history of viruses and the organisms that host them. How did the scientists do their research? Researchers studied where and when these pathogens were found in the ancient world, what species they infected, and how they changed to fit their mammalian hosts by looking at these fossils. To do this, they first looked through all of the databases of sequenced mammalian genomes for ERV-F. and then compared the sequences they found. For each genome with enough ERV-F sequence, they reconstructed the protein sequences that show how the virus infected the ancestors of that species. Then these sequences were used to figure out how ERV-FC-related viruses behave in nature and how they are related to each other in terms of evolution. The studies also helped the team figure out how the genes of these viruses have changed over time, which shows how they have adapted to different kinds of mammalian hosts. The researchers also found that these viruses often traded genes with each other and with other viruses. This suggests that genetic recombination was an important part of their ability to survive and spread over time. So, what have we learned? First, as we've already said, it seems like cross-species transmissions are more the norm than the exception in ERV-FC. The more we look, the more we find. Second, ERV-FC is a good genetic thief, and it often steals new genes from other viruses, at least twice, according to the data in this study. But they think they'd find a similar story if they looked at the rest of this group as closely as the study did at carnivores. It's tempting to think that these two things might be related. Did the carnivore FC1 viruses get a new gene give them the ability to bind to and enter different types of cells, allowing them to spread more widely? We don't know for sure, but we do know that virus receptor tropism can prevent transmission between species. For example, chickens appear to have become resistant to the mouse virus MLV by accumulating mutations in the receptors used by MLV's ENV. Lastly, what does this tell us about today's viruses and those that may threaten us in the future? To be honest, not much directly, but we think it raises an interesting question. How unusual is ERV-FC's proclivity for cross-species transmission? If it is unusually prone to crossing the species barrier, perhaps we can figure out what makes it so and keep an eye out for other viruses with similar abilities. And with that, we conclude today's video. What are your thoughts on the video? Tell us in the comments below. If you liked the video, give it a like and subscribe to the channel. Thank you for your time. We'll see you later.